So I'm going to quickly go through the homework. I'm going to go through 2 of the assignments myself, and then some others are going to review the homework they set. So the first piece of homework related to Josh Quick's talk, and this related to the software Primal Scheme. And to recap, this was the software that you use if you wanted to design a multiplex assay for doing viral sequencing. We talked about multiplex schemes for Ebola, and we talked about multiplex schemes for SARS-CoV-2, but we also pointed out that this could be used for many different other viruses, or even panels of bacterial genes or other microbial genes if necessary. So I will just quickly cover the answers here as a record for what the answers are and what you should have found. The idea here was that you would go and download a set of 10 Ebola genomes from GenBank, put those into a single FASTA file using a text editor, or you could do that directly through the NCBI website, and I put details of that on the Slack. Align the sequences together using Cluster Omega. Look at that alignment and just check that the genomes are roughly the same length. That was important because very long gaps in the alignment would be missed out from the scheme. So if you had 9 genomes that were the full length of the genome, about 20 kb, and you maybe had another genome that was only 10 kb, you would only design primer pairs for 10 kb, half the genome. It was important to put a divergent sequence set in where you didn't have too many sequences that were exactly the same or very nearly the same. And so point 5 asked you to look at the sequence divergence in Cluster Omega and check how divergent they were, and to look for sequences that were too similar that should be removed. And then finally, you should have run Primal Scheme on the sequences that you were happy to include in your analysis with 2 different amplicon lengths, 250 bases and 400 bases, and look at the output. So just to go through these steps in terms of the results that you should have had, it's possible using a link like the one pasted here to download all of the Ebola virus genomes by accession number with a link like that, where the accession numbers for each sequence are separated by commas. And GenBank actually makes it easy for you to first display that in FASTA format by clicking on FASTA under the little tab that usually comes up marked as summary. You can click FASTA and start seeing them in FASTA format, and then you can send them to a file in FASTA format that will end up giving you a text file with all 10 accessions contained within. You should have found the Cluster Omega alignment tool, multiple sequence alignment tool, just by searching for it on Google. And it's very simple to paste that file in or upload that file from your laptop, and make sure you choose that you've got nucleotide sequences and not amino acid sequences. And clicking on the results and looking at the percent identity matrix file, you would get a distance matrix like the one shown here. That's showing you the pairwise distances between sequences. On the diagonal, you see you're comparing each sequence to itself, so that will be 100%. And then for any given pair, you can see the distance. And so what you would have found is that, at least for the first 5 genomes, those genomes are quite different, maybe 2 to 3% sequence divergence. I think one of the questions was, which was the most different, which was the most diverged pair. More eagle-eyed people can probably pick that out. Pop it up in the chat if you've got an answer for that. Which of the 2, which pair are the most different? Let's see if anyone's going to say something in the chat. my chat box up. Oops. Okay. I can't get my chat box up. Here we go. Sorry, I can't find my chat box for some reason. Okay, I'll point that in a minute. And then, so the idea here is that you would look for sequences that were too similar, and I've put a black box around the sequences that I consider to be quite similar, all within about 1% sequence difference. Now, Josh recommends that you take a single version of those sequences, take a single example of those sequences, and remove the other ones. Because if you don't do that, if you have too many similar sequences in the input to primal scheme, the software will start to prioritize very similar sequences, and the scheme that you'll get out at the end will be very good for those particular types of Ebola virus, perhaps ones that all come from the same outbreak, but it will be less good at capturing sequence diversity from other Ebola viruses, making it a less useful scheme. So your results here, once you've taken those out, you can upload to the Primal Scheme website, which is primalscheme.com. And Josh asked you to try 2 different types of schemes. Initially, a 400 base pair scheme. So each of the amplicons in the primer pool will be about 400 bases when amplified. The distance between the primers is that long. And some statistics are given in terms of how well covered the genome is and in how many regions. So the first thing that you'll notice that the 400 base pair longer scheme will obviously produce fewer regions, fewer numbers of primer pairs are required to tile over that genome at 400 base pairs compared to if you use the 250 base pair scheme. So 61 primer pairs for 400 bases, 111 primer pairs for 250 bases. So shorter schemes would be more expensive because you would need more primers. But interestingly, the longer primer pairs give slightly better coverage of the genome here. The theoretical best coverage you'd get with this scheme at 400 bases is 98.86%, whereas at a shorter scheme, the coverage is 98.58%. And not just that, with the shorter scheme, you end up with a couple of gaps that are difficult to find primer pairs for. And the reason for this is simply that as the number of primers that you require goes up, the chances of finding a really good primer sequence, oligo sequence that will work well with the PCR that we recommend, the PCR conditions we recommend for this protocol, the chances go down. So it's easier to design longer schemes. The flip side of that is that longer schemes, if you do end up with a primer dropout, will result in you losing more of your genome. So that's the kind of trade-off there. Okay, I'm just going to stop the share because I just need to find my chat box, which, there we go, I've got it now. Okay, and I can see John Juma says that the largest divergence was 3.4%. Let's just go back to the presentation, just check on that. 3.4%. So that's going to be this one where we on the top row where it says 96.64%. I think that's probably right. So excellent. Okay, I need to move quickly on because we don't have a huge amount of time to review the homework. So the second piece of homework was to look at the ARTIC bioinformatics pipeline, and hopefully give us a thumbs up if you managed to run this through the MyBinder, which is a very easy way of doing things, I think. Anyone managed to do this? Not seeing much in the way of thumbs up, but And so yeah, Will managed it. John managed it. That's good news. Okay. Sorry. So John also said, can we remove sequences KF 113528 versus KC 24800 because they're 99.62? Yes, you could do that. Okay. So Will set a bunch of questions here about the output of the Arctic bioinformatics pipeline. So the questions he asked were, how many amplicons were classed as low coverage and what was the reported coverage for these? How many variants were identified that were subsequently marked as fail, perhaps because they were low quality? How many variants were ultimately incorporated into the consensus genome? And how many Ns are in the consensus genome and why might they be there? So there's lots of different ways you could have done, could have got the answers here, but the easiest way to answer the first question, which is how many amplicons were marked as QC fail, maybe post up your answer in the chat if you know the answer to this one. But the way you would do this is to look at the coverage plot from the output of multi-QC, which is a tool that will give you a web page coverage report from the output of the Arctic pipeline. Not seeing anyone in the chat posting the answer. The answer here was that one amplicon was marked as QC fail, and that is amplicon 74. And you can see 74 on this chart just drops below the dotted red line, and the coverage of that amplicon was 34-fold. So in the multi-QC report, anything below 50 times coverage is marked as a coverage fail. Having said that, it would still be incorporated into the final consensus genome. It would need to be below 20 to be excluded completely. In terms of how many variants were detected, there were 9 variants in total that were detected and passed the quality filters in order to make it into the eventual consensus sequence, and those are listed here. If you were looking in detail, you would have seen that the VCF file actually contained a total of 13 variants. Four out of those 13 are duplicated. So in positions where the variants are detected in an overlap between 2 amplicons, those variants are reported twice. So they're included, but they're deduplicated. So 13 goes down to 11. And 2 positions are reported as variants, but with very low quality. Those are both single base insertions, and those were excluded because they don't meet the quality threshold. So 13 reported goes down to 9. And finally, we asked how many N characters were in the consensus file. And there are a few different ways you could have done that, but you could have simply opened the consensus file and just searched for Ns with a text editor. You could have written a little bit of Python script using the example Python that we gave you in the IPython notebook, in which case you would have got the result of 121 Ns, and those represent the extreme ends of the genome that are not covered by the primer scheme. And you could have also looked at the coverage mask file and seen where the Ns came from. So a few different options there. If none of this makes sense, I would encourage you to go back to the MyBinder website after the course and look at this in much more detail. Okay, in the interest of time, we'll move on again to the next bit of homework, which was set by Nabil on bioinformatics gotchas. And I'm going to ask Nabil to present very quickly the results of this, if that's all right, Nabil.
Yep. Okay, so this is a simple, more theoretical exercise for everyone, just getting you to think about data integrity and there was a sample table from a sequencing site which had a bunch of different records there, and there was at least one mistake in each of the rows. Hopefully you spotted them all. It's written there underneath the table in the for the answers. For instance, the sample ID in the first one, the site's name is NOGG, N-O-G-G, not N-O-F-F. So the second row, the collection date is well before it could possibly be. It appears to be the birth date of the patient rather than the actual collection date. The third one has a collection date that's in the future and a typographical error. The next one has an error with the country and the patient age. Hopefully you noticed that 121 would make them the second oldest person on a living person. So that's quite unlikely that that's the case. The one after that just has some extra characters added on, just someone just hit the keyboard at the wrong time. And then the last one has the sequencing technology is the Illumina MinION, which does not exist. It should be the Oxford Nanopore MinION, or it might have been the Illumina NextSeq, but they sort of mixed it up somewhere. So that's the first one, pretty easy. The second part of this question was to come up with ways to check these in future. I think it's on the next slide, Nick. Yeah, so for instance, how do you make sure that the sample ID in future doesn't come up as NOF? There is a specific format. If you look at each of the sample IDs, it seems to follow a specific format of the site name, a hyphen, a digit, a capital letter, and then 2 additional digits. And if clever among you might have come up with a regular expression, that's my suggestion. You might have something else. If you do, put it in the chat. Then type is always a beta coronavirus. You should have restrictions on the date, restrictions on the country and the host and the sex. That's easy to just have a list of possibilities for that. Patient age should have a specific range and should always be a number. And then there should be a fixed list of sequencing technologies, and that would catch all of the errors from in the table. Then there was a second question after that. I think it's on the next slide, where this was going back to when I was talking about sample identifiers, and I gave you a list of sample identifiers which meet different requirements. So a good sample identifier should be human readable, computer readable, and unique. And each one of these only partially meets those requirements. So the first one is basically an MD5 checksum. It's long, it's unique looking, probably computer readable, but it's not human readable. It's not something you want to have in lab books and transcribe and explain to someone over the phone. The second one is the sample 001, which is human readable, it's computer readable, but it's probably not unique. Many people are probably going to adopt that sort of naming and you're going to get confusion. The one after that, it has a comma and a colon in it, which is a little problematic for some computational scripts because commas and colons are often used as delimiters. So like a comma delimited text might have a struggle with this. So it's not particularly computer readable. And then the last one has some Unicode characters, which may cause a problem for particular software. So again, that's not computer readable. So you might have come up with different reasons or a slightly different spread for each of the questions. And I'm happy to discuss that on the Slack or the chat if you want to go any further. But that was it for me. It was a pretty simple exercise.
Simple, but I think a really good exercise, and surprising how many people fall foul of some of these rules, even quite experienced labs, I think. So thank you very much for that one. We're now I'm gonna hand over to Verity and Anya to present the results and the answers for the genomic epidemiology homework. Are you okay to take over and share your screen?
That's no problem. I'll share now. You might see yourself for a second, Nick. That's okay. Okay, so you can all see that, all right? It's quite a simple exercise. If you tried it out, you'll realize that. But it's just to take you through assigning some lineages with the Pangolin web app, with an example set of sort of this outbreak schema. So this is just a recap. Verity took you through this yesterday, what Pangolin is, and where the sequences and the metadata are. So this was all made up data, and they're real sequences, but all the metadata is just completely fabricated. So you had this example. There were some metadata cases of travel history of where patients might have come from, the sort of mixture of patients and healthcare workers across two different kinds of wards, and then the healthcare workers are not assigned particular wards. So you have your sequences. It's very simple. You drag your multi-FASTA file into the web app, and all going well, you run the analysis and should get a result that looks a little bit like that. So you see that the third and fourth sample actually failed QC. So you click into it, you can get a little bit more information, and you can see we've actually got two different kinds of lineages in the sort of other examples. So if we go back to our metadata and have a look at what lineages were what, you can see we've got lineage B and lineage B.1.1.26. So it's quite a simple answer, but it's a kind of powerful example of what fine scale lineages can do. So you can see that sample 5 is on the same ward as samples 6, 7, 8, and 9, but it's actually got a different lineage. So the likelihood is that it would have a different source of infection. The two samples that failed, you might need to go back and resequence if you have more of that sample. But if there's too many ends or not enough data to give reliable answers, then you wouldn't want to be using those sequences anyway. So so we can say that sample 5 isn't linked to the rest of the samples on ward 6. We can see that 5 and 10 are the same lineage, but beyond that, we can't make assumptions. We would need to have additional information about maybe what lineages were circulating in the area at the time. We can't infer any sort of direct transmission with that. We would want to maybe supplement that information with some more epi data. We can see again that one is on the same, sample 1 is the same lineage as on another ward, but both of those individuals have traveled from the US. So we have all these pieces of information. The lineage assignment is just one more piece of information. So we can't make assumptions about whether those cases are linked. They are on different wards. If you had information about what lineages were circulating in the US at the time, you might be able to say whether or not that's a common lineage that's in the US, and you sort of can use all these different pieces to piece together the jigsaw. So yeah, so that's where you tried it out. So hopefully you kind of made the same conclusions as we just went through there. So again, it's really, really simple. I'll try and stop this share, or if, there you go. Yeah, so very simple, but quite a powerful piece of information to have.
That's great. Give us a thumbs up, or if you're anyone that managed to do the Pangolin lineage tutorial.
Or if there's anyone who has questions about sort of the interpretation of what that actually means, and you know, the sort of hierarchy of it or anything like that, we can talk about it in the session later, or if you want to shout out now, that's fine too. So.
That's great. I did see quite a few thumbs up there. That's nice. Obviously, hopefully, if you haven't done that, it will inspire you to go and do it, because that is a really easy, quick way of, as Anya says, ruling out a transmission at the very least. It's much more work to rule stuff in, but to rule stuff out, being in different lineages is a very, very rapid way of identifying. Was Verity going to present the Civic output as well?
Yes.
Okay, lovely. Over to you, Verity.
Okay, so I'm going to just share this. So can you see the Civic report?
Yep.
Okay, great. So as Anya says, we've got these two kind of groups of sequences, and some of the transmission we can rule out and some of it we can't. So we want to dig into a few other things. So you can also run Civic on these sequences. And remember, we were also interested in one of the sequences came from a doctor who had had some friends visit from other parts of Scotland. So we wanted to find out if those friends were also maybe involved in this hospital outbreak that we were concerned about. So this is what a Civic report looks like. Hopefully, if you didn't manage to run Civic, you were still able to look at the report that we provided. It starts off with some information about the analysis. So the first question that I put down to think about is why a couple of the sequences were missed out, and it's just here they it was actually, I think, the same ones that were picked up by pangolin, but just to show that if you didn't do the pangolin one, Civic would also pick them up as well. So yeah, way too many ends and just too short. So that means that we can't do accurate phylogenetics with them. And then this first table here are the queries that were matched to the background database. So those are the two friends that we didn't have sequences for because they'd already been sequenced. So we didn't need to re-input that data. And then here are 4 sequences that we were interested in the ward. So yeah, and then we've got the trees of interest. So Civic will go into the big tree, as Anya was saying yesterday, and pull out the relevant catchment area of the sequences of interest. So that's what's happening here. So this is a tree showing 2 of the sequences and information about them, and I'll just go into that in a second. You can see that this sequence is actually in a different catchment tree, so it's just in a different part of the big tree. And each of these groups of figures is shown per tree. And then the additional information that we have here for each tree is we have colored the tips by relevant information. So this is by the healthcare worker status. So whether they were a member of the community, which is like the friends, whether they are a healthcare worker, so it's like a doctor or a nurse or whatever, or the patient, and then also the ward that they were in. So that just to show that on the tree, trees are quite a nice way of visualizing data like that. And then this is a timeline. The legend is a bit small, but the brown dots are symptom onset and the blue dots are sample dates. That can help us, again, give weight to different hypotheses about transmission. And then this is a snippet graph. This is a package that Anya's developed, which is actually independent of Civic. You can just use it in your normal day-to-day code, and it zooms in on the different parts of the alignments that have the relevant SNPs that are on the branches leading up to the sequences of interest. And that kind of helps us if sometimes it looks like there are SNPs on branches, but it's actually missing data or ambiguous data. So it just highlights sometimes that we need to be a bit careful about how we interpret it. So firstly, we were interested in the friends from other parts of Scotland. That's quite a nice, easy one here. We can see that, so this is the doctor, this 10, EDB10. We can see that one friend is in this tree and one friend is in this tree. So they're really unrelated to each other. We can rule out transmission. We can just say that these 2 friends both got independently infected from other parts of the community. So that's nice. We can just rule that out. We don't need to worry about them. The next thing is whether the patient and the doctor are connected. This is a bit more tricky. So we already knew they were part of the same pangolin lineage, but we wanted to dig in a bit more and see if they were more closely related. So they are identical. They're on a very small polytomy. So that gives some evidence in the kind of pro that they might be connected. Another pro piece of evidence is that their timelines overlap in a way that is biologically plausible. So the incubation time for SARS-CoV-2, I think, is between about 2 and 14 days, and you're infectious for about 48 hours before you start developing symptoms. So the patient here developed symptoms 5 days after the doctor, so that is in line with the time in which the doctor was infectious. So we've got like a few pieces of evidence here that could mean that the doctor or the patient infected each other. But the thing is we can't say for sure. So to be sure about this, we would need to get some other pieces of context, like, is it even possible that the doctor could have infected the patient? Were they in contact with each other? Is this a patient who's been in the hospital a long time and caught COVID in the hospital, or were they just completely unconnected in terms of their like social contacts? We could do it a little bit more genomic data, and with Civic, you can ask it to show you more of the tree. So that would be quite useful here. We can see that the nearest sequence is like a SNP or so away. So yeah, they are a bit different to the closest sequence, but there could be unsampled cases here, which we're not seeing, which would help us to distinguish that. Yeah, I think that's kind of the main thing. So basically, yeah, it looks like they could be connected. We can't say for sure. We can't say directionality because the timing would just work with the patient infecting the doctor. So there's, yeah, it's complicated basically, and probably what you would do is still try and tighten up those IPC things just to be on the safe side, just in case, because it is plausible that the doctor infected the patient or vice versa. We can't rule it out. And the final thing, just whether there was infection between wards. So the other patients are down here. So they're on the same tree, but they are a SNP apart. So it's probably reasonably unlikely that they connected in a transmission chain for SARS-CoV-2. If you were looking at a different virus which had a faster mutation rate, you might interpret this differently, but it's quite unlikely that they infected each other here. Although, again, we would want to look at context. If one of them coughed on the other one 3 days before they got symptoms, then that would give us some evidence that they might have infected them. So I think, yeah, I think that's everything for the Civic one. If anyone has any questions on any of the bits, can obviously ask now or later or anything like that. Sorry, some of the installation problems were happening. We haven't seen some of those before. They were strange.
All right, brilliant. That's fantastic, Verity. Thank you. As you say, we will take questions on all of these parts of the homework and anything else on your mind after the next talk in the Q&A panel session. So if you do have questions and concerns, we can address those specifically.